Assalamu alaikum class. Today we will talk about gluconeogenesis. As the name says, gluconeogenesis is the biosynthesis of new glucose, not the glucose from glycogen, the new one. This process is frequently referred to as endogenous glucose production as well, or we can abbreviate it as EGP. Some tissues, such as the tissues of the brain, red blood cells, kidneys, lens, cornea of the eye, testes, and exercising muscles require a continuous supply of glucose as a metabolic fuel. Liver glycogen, which is an essential source of glucose, can meet these needs only for 10 to 18 hours in the absence of dietary intake of carbohydrates. During a prolonged fast, however, hepatic glycogen stores are depleted and glucose is formed from precursors such as lactate, pyruvate, glycerol, which is actually derived from the backbone of triglycerols, and alpha keto acids. The brain alone requires about 120 grams of glucose each day, which is more than half of all the glucose stored as glycogen in the muscles and liver. However, the supply of glucose from these stores is not always sufficient. Between meals and during longer fast or after a vigorous exercise, glycogen is depleted. For these times, organisms need a method for synthesizing glucose from non-carbohydrate precursors. This is accomplished by a pathway which is called gluconeogenesis or formation of new sugar. This converts pyruvate and related 3 or 4 carbon compounds to glucose. Gluconeogenesis occurs in all the animals, plants, fungi, and even in the microorganisms. The reactions are essentially the same in all the tissues and all the species. The important precursor of glucose in animals are the 3 carbon compounds such as lactate, pyruvate, and glycerol, as well as certain amino acids. In mammals, gluconeogenesis takes place mainly in the liver and to a lesser extent in the renal cortex. The glucose produced passes into the blood to supply other tissues. After a very heavy exercise, lactate produced by the anaerobic glycolysis in the skeletal muscles return to the liver and it is converted into glucose, which then moves back to the muscles and it is converted into glycogen. It's a kind of circuit which is called the Cori cycle. The formation of glucose does not occur by a simple reversal of glycolysis because the overall equilibrium of the glycolysis strongly favors pyruvate formation. Instead, glucose is synthesized by a special pathway which is gluconeogenesis and that requires both mitochondrial and cytosolic enzymes. During an overnight fast, approximately 90% of the gluconeogenesis occurs in the liver, with the kidneys producing about 10% of the newly synthesized glucose molecules. However, during prolonged fasting, the kidneys become major glucose producing organs and contribute an estimated 40% of the total glucose production. Now, in case of plant, glucose is the product of, as a result of photosynthesis, glyceraldehyde 3 phosphate is produced. And this is then converted to starch and stored in the chloroplast, or it is converted to glucose and sucrose and exported to the other tissues for starch storage. In plant seedlings, the stored fats and proteins are converted by paths that include the gluconeogenesis to the disaccharide sucrose for transport throughout the developing plant. Now, glucose and its derivatives are the precursor of the synthesis of plant cell walls and nucleotides and coenzymes and a variety of essential metabolites. In many microorganisms, gluconeogenesis starts from simple organic compounds which are 2 or 3 carbon long, such as acetate, lactate, or propionate, and in the growth medium when they are added. Now, although the reactions of gluconeogenesis are the same in almost all the organisms, the metabolic context and the regulation of the pathway differs from one species to another and from one tissue to another. And in this series of lecture, we are going to focus on gluconeogenesis and as it occurs within the mammalian liver. Now, here you see that carbohydrates are actually synthesized from simple precursor, as you can see in this diagram. Now, the pathway from the phosphoenol pyruvate to glucose 6-phosphate is common to the biosynthetic conversion of many different precursors for carbohydrates in animals and plants. Plants and there are many photosynthetic bacteria as well. They are uniquely able to convert carbon dioxide to carbohydrates. Now, here is the process of glycolysis and gluconeogenesis at the same time. And here you can see the opposing pathways of glycolysis and gluconeogenesis in the liver of a rat. Now, gluconeogenesis and glycolysis are not identical pathways which are running in like opposite direction, although they do share several steps, as you can see in this figure. Now, 7 of the 10 enzymatic reactions of gluconeogenesis are the reverse of glycolytic pathway or the glycolytic reaction. However, these three reactions of glycolysis are essentially irreversible in vivo and cannot be used in the synthesis of new glucose through gluconeogenesis, which is the conversion of glucose to glucose 6-phosphate by like hexokinase, the phosphorylation of fructose 6-phosphate to fructose 1,6 bisphosphate by the enzyme phosphofructokinase 1, and the last, the conversion of phosphoenol pyruvate to pyruvate by the activity of the enzyme pyruvate kinase, as you can see in the reactions which I have like marked in red boxes. In cells, these three reactions are characterized by a large negative free energy change, which is G gives free energy, as you remember, whereas other glycolytic reactions have a change in the free energy or gives free energy, which is nearly zero. In gluconeogenesis, the three irreversible steps, which I have shown here in red boxes, they are bypassed by a separate set of enzymes, which are catalyzing the reactions that are sufficiently exergonic to be like effectively irreversible in the direction of glucose synthesis. Thus, both glycolysis and gluconeogenesis are irreversible processes in cells. In animals, both pathways occur like largely in the cytosol. Actually, their reciprocal and their regulation is actually coordinated. Now, separate regulation of the two pathways is brought about through controls exerted on the enzymatic steps, which are unique to each pathway. Now, we will begin by considering the three bypass reactions of gluconeogenesis, and you must keep into your mind that the bypass actually refers throughout to the bypass of the irreversible glycolytic reactions. Now here you see three different substrates which are used for the synthesis of new glucose by the process of gluconeogenesis. We have glycerol, we have lactate, and we have amino acids. Now, the gluconeogenic precursors, that are the molecules that can be used for the production of or synthesis of new glucose, they include intermediates of glycolysis and the Krebs cycle or the TCA cycle. We have glycerol, lactate, and acids, which are these amino acids are actually the alpha keto acids, which are obtained from the transamination of like glucogenic amino acids and the most important glucogenic precursors. Glycerol is, is released during the hydrolysis of triglycerol in the fat tissues, or we also call them the adipose tissue, and it is delivered by the blood to the liver. Glycerol is actually phosphorylated by the glycerol kinase enzyme, and it forms glycerol phosphate, which is oxidized by glycerol phosphate dehydrogenase to like dihydroxyacetone phosphate, which is also an intermediate of glycolysis, if you remember. Lactate, on the other hand, is released into the blood by exercising skeletal muscles. And if you remember in the process of glycolysis that I explained you, that when there is less supply of oxygen, pyruvate is converted into lactate by the process of fermentation. Now, this lactate, which is released into the blood by the exercising muscles and by the cells that do not have a mitochondria, such as the red blood cells, In the Cori cycle, like I told you, the process in which the glycolysis and gluconeogenesis, the process in which glucose is being converted into glycogen and then back into glucose, the blood-borne glucose is converted by the exercising muscles to lactate, which diffuses into the blood. This lactate is taken up by the liver and it is reconverted to glucose, which is then released back into the circulation. Now, if we look at the alpha keto acids or the amino acids as a substrate for the source of gluconeogenesis, these amino acids are actually derived from the hydrolysis of tissue proteins and they are the major source of glucose during fast, like we are fasting nowadays. So the amino acids are being used up. Now, alpha keto acids such as alpha ketoglutarate, they are derived from the metabolism of glucogenic amino acids. These alpha keto acids can enter into the TCA cycle to form oxaloacetate, if you remember the cycle, which is a direct precursor of the phosphoenol pyruvate. Now, acetyl coenzyme A and the compound that give rise to only to acetyl coenzyme A, for example, the acetoacetate and the amino acids such as lysine and leucine, they cannot give rise to a net synthesis of glucose. This is due to the irreversible nature of pyruvate dehydrogenase reaction, which converts pyruvate into acetyl coenzyme A. Now let's look at the conversion of pyruvate to phosphoenol pyruvate. Okay, the first bypass reaction in gluconeogenesis is the conversion of pyruvate to phosphoenol pyruvate. We are, you can say, looking at the process of glycolysis starting from the last reaction, right? So this reaction cannot occur by the reversal of the pyruvate kinase reaction of glycolysis, and since it has a large negative standard free energy change and it is irreversible under the conditions prevailing in intact cells. Like instead, the phosphorylation of pyruvate is achieved by a roundabout sequence of the reaction in the eukaryotic cells, which require enzymes in both the cytosol as well as in the mitochondria. Pyruvate is first transported to the cytosol into the mitochondria, and it is generated from alanine within mitochondria by the process of transamination, in which the amino group is removed from the alanine. When it is removed, pyruvate is left behind, and it is added to ketocarboxylic acid. Then this pyruvate carboxylase, which is, a mitochondrial enzyme that requires the coenzyme biotin, converts pyruvate to the oxaloacetate. So the reaction that happens is that pyruvate, it combines with carbonic acid along with the ATP molecule, and it results in the production of an oxaloacetate along with the adenosine diphosphate molecule and an inorganic phosphate. Pyruvate carboxylase is one of the several carboxylases that requires biotin. Other include acetyl coenzyme A carboxylase, propionyl coenzyme A carboxylase, and methyl carotenoyl coenzyme A carboxylase. You see, these are all carboxylases. Now, the reaction involves biotin as a carrier of activated carbonic acid or HCO3. The reaction mechanism is shown in the figure. As you can see, that pyruvate carboxylase is first regulatory enzyme in the gluconeogenic pathway, requiring acetyl coenzyme A as its positive effector. Acetyl coenzyme A is produced by fatty acid oxidation as well, you know that, and its accumulation signals the availability of fatty acids as fuel. Now, because of the mitochondrial membrane has no transporters for the oxaloacetate before its export to the cytosol, the oxaloacetate formed from pyruvate must be reduced or converted into malate by mitochondrial enzyme, which is called as malate dehydrogenase. And during this process, NADH molecules are used up. So the net reaction is that oxaloacetate combines with the NADH molecule along with the proton in a reversible manner and produces malate. It's L isomer along with NAD molecule, which is now in oxidized form. Now, the standard free energy change for this reaction is quite high, but under physiological conditions, like very low concentration of oxaloacetate, the Gibbs free energy or change in the free energy is going to be almost zero. And the reaction is therefore readily reversible. Mitochondrial malate dehydrogenase functions in both gluconeogenesis as well as in the citric acid cycle. But the overall flow of the metabolite in the two processes is in opposite direction. Now, malate leaves the mitochondria through a specific transporter in the inner mitochondrial membrane, and in the cytosol, it is again reoxidized to produce oxaloacetate. And since oxaloacetate is produced, we have cytosolic NADH molecules and malate. They are then produced. So malate, when it combined with the NAD molecule, oxaloacetate is produced along with the regeneration of NADH molecule along with protons in the cytosolic region. The oxaloacetate is then converted into phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxylase. This ion, I think I have already told you that this carboxylase requires magnesium ions. So this is a magnesium dependent reaction, and this also requires a GTP as the phosphoryl group donor. So the overall reaction is that oxaloacetate combines with a GTP molecule, producing a phosphoenol pyruvate along with carbon dioxide, and therefore GTP is converted into GDP. Now, this reaction is reversible under intracellular conditions. That's the formation of one high energy phosphate compound, which is phosphoenol pyruvate, and it is balanced by the hydrolysis of another, which was GTP molecule. The carbon dioxide, which was added to the pyruvate in the pyruvate carboxylase step, it is the same molecule that is lost in the phosphoenol pyruvate carboxykinase reaction. This carboxylation or decarboxylation sequence represents a way of activating pyruvate, and this activated pyruvate, which is in that the decarboxylation of the oxaloacetate is facilitates the production of phosphoenol pyruvate. Now here you see the conversion of pyruvate to phosphoenol pyruvate. In this diagram, you can see the overall process of conversion of pyruvate to phosphoenol pyruvate, and you see here the activation of and the transfer of carbon dioxide to pyruvate, and then it is followed by the transport of oxaloacetate to the cytosol and its subsequent decarboxylation. Now, this phosphoenol pyruvate that is generated in the mitochondria is transported to the cytosol, as I told you, by specific transporters. Whereas that generated in the cytosol, it requires the transport of oxaloacetic acid from mitochondria to the cytosol. Now, however, oxaloacetic acid is unable to directly cross the inner mitochondrial membrane. It must first be reduced to malate by mitochondrial malate dehydrogenase, like I told you in the previous slide. Now, malate can be transported from mitochondria to the cytosol, where it is going to be again reoxidized to oxaloacetate by the enzyme, which is cytosolic in nature, malate dehydrogenase. And during this process, you see that a NAD molecule is being reduced. Now, the NADH produced is used up in the reduction of the enzyme of the product 1,3-bis phosphoglycerate to glyceraldehyde 3-phosphate, which is also a common step between the process of glycolysis and gluconeogenesis, as you can see here in this diagram. Now, oxaloacetate is decarboxylated and phosphorylated to phosphoenol pyruvate in the cytosol by the phosphoenol pyruvate carboxylase, and the reaction is driven by the hydrolysis of guanosine triphosphate GTP, as you can see here. Now, the combined actions of the pyruvate carboxylase and phosphoenol pyruvate carboxylase kinase provide an energetically favorable pathway for the conversion of pyruvate to phosphoenol pyruvate. Then this phosphoenol pyruvate is acted upon by the reactions of glycolysis running in the reverse direction until it becomes fructose 1,6-bisphosphate. Now we have the diagrams which are actually explaining the dephosphorylation of fructose 1,6-phosphate. So, the hydrolysis of fructose 1,6 bisphosphate by the enzyme fructose 1,6 bisphosphatase bypasses the irreversible phosphofructokinase 1 reaction, and therefore it provides an energetically favorable pathway for the formation of fructose 6 phosphate, right? The figure that you can see on the left side. Now, this reaction is important regulatory site for gluconeogenesis. There are two types of regulations going on at that time. First of all, regulation by the energy level within the cell. When fructose 1,6 bisphosphate is inhibited by the elevated level of adenosine monophosphate or AMP molecules, which signal an energy poor state in the cell. Conversely, high levels of we have high levels of ATP and low concentration of adenosine monophosphate. It stimulates gluconeogenesis, which is an energy requiring pathway. Second regulation is the regulation by fructose 2,6 bisphosphate. Now, fructose 1,6 bisphosphatase, which is found in the liver and kidney, it is inhibited by fructose 2,6 bisphosphate, and it produces a allosteric effect whose concentration is like influenced by the level of circulating glucagon. As you can see in this figure, the effect of elevated glucagon on the intracellular concentration of fructose 2,6 bisphosphate in the liver. And here you see there is an abbreviation written PF and K, PFK2, which is phosphofructokinase 2. And we also have FBNT 2, which is actually fructose bisphosphate 2 enzyme. So now we have dephosphorylation of glucose 6 phosphate. Now this is the third bypass, and it is the final reaction of gluconeogenesis, which is the dephosphorylation of glucose 6 phosphate to produce glucose. Now reversal of the hexokinase reaction would require like phosphoryl group transfer from glucose 6 phosphate to adenosine diphosphate molecule, producing a ATP, and energetically this is going to be a like unfavorable reaction, right? The reaction is actually catalyzed by glucose 6 phosphatase enzyme, and it does not require synthesis of ATP. And it is a simple hydrolysis reaction, and it involves hydrolysis of phosphate ester, in which a glucose 6 phosphate it combines with water and results in the and each glucose molecule is going to like along with an inorganic phosphate, it is going to like produce energy of about is going to require energy of about like 13.8 kilojoules per mole. Now, the magnesium activated enzyme is found in the luminal side of the endoplasmic reticulum of the liver cell or hepatocytes and the renal cells. Muscles and brain tissue do not contain this enzyme, and therefore they cannot carry out the process of gluconeogenesis. Now, glucose produced by gluconeogenesis in the liver or the kidney, or if it is ingested in the diet, it is delivered to the brain and muscle through the bloodstream. Hydrolysis of glucose 6 phosphate by glucose 6 phosphate bypass the reversible hexokinase reaction and provides an energetically favorable pathway for the formation of free glucose. Liver and kidney are the only organs that release free glucose from glucose 6 phosphate. Now, this process actually requires two proteins. We have glucose 6 phosphate translocase, which transports or translocates glucose 6 phosphate across the endoplasmic reticulum membrane, and the endoplasmic reticulum enzyme, and its name is glucose 6 phosphatase, which is found only in the gluconeogenic cells. And it removes the phosphate, producing free glucose, and specific transporters are responsible for releasing free glucose and phosphate back into the cytosol for glucose into the bloodstream. Now, muscles also lack glucose 6 phosphatase, and therefore muscles glycogen cannot be used to maintain blood glucose levels. Now, when we look at the regulation of the gluconeogenesis, I have this reaction written, which is actually representing the sum of the total reaction taking place for the conversion of pyruvate into glucose. So here you see that the sum of the biosynthetic reactions leading from pyruvate to free blood glucose is for each molecule of glucose formed from pyruvate, which is like six high energy phosphate groups are actually required, from the ATP and two from the GTP for this reaction to take place. Now, in addition to two molecules of NADH are required for the reduction of the two molecules of 1,3-bis phosphoglycerate. So clearly from this equation, it is like not simply the reverse reaction or equation of the conversion of glucose to pyruvate like by glycolysis, which requires only like two molecules of ATP. Here you can see that we are using up more energy. So the synthesis of glucose from the pyruvate is a relatively expensive process, and much of this high energy cost is necessary to ensure the irreversibility of gluconeogenesis. Under intracellular conditions, the overall free energy change for glycolysis is around 63 kilojoules per mole. Now, under the same condition, the overall change in the free energy of the gluconeogenesis is 16 kilojoules per mole. Thus, both glycolysis and gluconeogenesis are essentially irreversible processes in the cells. So, if glycolysis, which is the conversion of glucose to pyruvate, and gluconeogenesis, which is the conversion of pyruvate to glucose, were allowed to proceed simultaneously at high rate, the result would be the consumption of ATP and production of heat. For example, we have phosphofructokinase 1, and there's another enzyme which is fructose bisphosphatase 1. It catalyzes opposite reactions. As you can see in this reaction, ATP is being used up and we are producing by the help of this enzyme ATP along with fructose 1,6 bisphosphate, and this fructose 1,6 bisphosphate is undergoing a hydrolysis reaction producing fructose 6 phosphate and a inorganic phosphate, and the sum of the two reactions that ATP along with the water molecule being used up, and you see that ADP along with an organic phosphate, and heat is being produced. You see here. Now, the sum of these reaction is that ATP along with the heat is being produced. Now, these two enzyme reactions and a number of other in the two pathways are, as I told you, they are regulated, and they are regulated allosterically and by covalent modifications. The most important or common covalent modification that we have witnessed during the study of glycolysis, TCA cycle, transport chain, and now gluconeogenesis is actually phosphorylation. The pathways, they are regulated so that when the flux of glucose through glycolysis goes up, the flux of pyruvate towards glucose goes down, and vice versa. So, at the end of understanding these three reactions, which are irreversible in the process of glycolysis to produce glucose by the process of gluconeogenesis, that is production of new glucose, we have some points that we can summarize as a result of the discussion that we have done today. In this summary, you can see the sequential reactions in gluconeogenesis starting from pyruvate, and some of the reactions are in red, the other ones are black. The red ones are the reactions that are irreversible, while the black ones are the reversible. And the word X and 2, which is written on the right side of the slide, it is the times this reaction is actually taking place. So the bypass reactions, as I told you, they are in red. All the reactions, other reactions are reversible, and they are the steps of glycolysis, and they can be counted twice because 2 to 3 carbon precursors are required to make a molecule of glucose. Now, the reactions required to replace the cytosolic concentration of NADH molecule consumed in the glyceraldehyde 3-phosphate dehydrogenase reaction, which was the conversion of lactate to pyruvate in the cytosol, or the transport of reducing equivalent from mitochondria to the cytosol in the form of malate, they are not considered in this summary. So, the overall summary of the gluconeogenesis is that First of all, gluconeogenesis is a ubiquitous multi-step process in which pyruvate or related three-carbon compound, for example, lactate or alanine, it is converted into glucose. Seven of the steps in this gluconeogenesis are catalyzed by the same enzymes that were used up in the process of glycolysis, and these are the reversible reactions. Three irreversible steps in the glycolytic pathways are bypassed by reactions catalyzed by gluconeogenic enzymes. First reaction is the conversion of pyruvate to phosphoenol pyruvate with the help of enzyme oxaloacetate through oxaloacetate catalyzed by the enzyme pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Second reaction is dephosphorylation of fructose 1,6-bisphosphate by the phosphatase enzyme FB phase 1, and which is abbreviated like this. And the third reaction is dephosphorylation of glucose 6 phosphate by glucose 6 phosphatase. The formation of one molecule of glucose from pyruvate requires 4 ATPs, 2 GTPs, and 2 NADH, and therefore it is quite expensive. In mammals, gluconeogenesis in the liver and kidney provides glucose for use by the brain, muscles, and erythrocytes. Pyruvate carboxylase is stimulated by acetyl coenzyme A, increasing the rate of gluconeogenesis when the cells already have adequate supplies of other substrates, for example, fatty acids for the production of energy. Animals cannot convert acetyl coenzyme A derived from the fatty acid into glucose, whereas plants and microorganisms can do that. Glycolysis and gluconeogenesis at the end are reciprocally regulated to prevent wasteful operations of both of the pathways at the same time. So here we end our process of gluconeogenesis.